You are listening to the Effective Statistician podcast, the weekly podcast with Alexander Schacht and Benjamin Pieske, designed to help you reach your potential, lead great science, and serve patients without becoming overwhelmed by work. Today we are talking about paedriatrics and how we can use extrapolation to help have data or evidence for a pediatric submission. And here we have Andrew Thompson from the regulatory side here. So stay tuned for this really good discussion with him. And now some music. Understanding about a pediatric submission is really important. Maybe you have kids yourself. I do have kids. And there's always a lack of treatment in that area. So I think as an industry, we need to become better in serving this population. Of course, there's certain diseases that, there's really no kind of pediatric indication, but very often there is. And so this episode will help you understand what you can do to get evidence through extrapolation for this population. I'm producing this podcast in association with PSI, a community dedicated to leading and promoting the use of statistics within the healthcare industry for the benefit of patients. Join PSI today to further develop your statistical capabilities with access to the video-on-demand content library, free registration to all PSI webinars, and much more. The reduced rate is only £20 for non-high income countries and £95 for high income countries. And of course, there's the PSI conference in Gothenburg coming up. So head over to psiweb.org to learn about the conference and all the other PSI activities. And hopefully see you in Gothenburg. Welcome to another episode of the Effective Statistician. Today I'm really happy to have Andrew Thompson here, who is really an expert in the topics that we are talking about. And not only that, he's also coming from the regulatory side of things, which I think is maybe the first time I have someone from the EMA or the FDA on my podcast. So I'm really honored about this. Hi, Andrew. How are you doing?
Hi, Alexander. I'm very well, thanks.
Very good. Before we dive into the topic, maybe you can speak a little bit about your career and why you actually went into biostatistics in the first place.
Yeah, sure. So I started like many statisticians doing maths at university, and I found I wasn't particularly good at either pure or applied maths, but somehow I had a knack of being okay at statistics. And in my final year, there was an advert on the screen in one of my stats lectures for an MSc place in statistics with applications in medicine, which would be funded. And I thought that sounded like an interesting thing to do for a year, and the funding certainly helped, and never really looked back since. And I started off wanting to be an academic, and I worked at Imperial College and then did a PhD at London School of Hygiene. And then towards the end of my PhD, I wasn't so sure that I wanted to be an academic after all, and I looked at the alternative options out there for me. And at that time, it just so happened that the MHRA were recruiting for statistical assessors. And I applied, and I was very fortunate to be offered a position. And so I started work doing statistical assessment, looking at dossiers across all therapeutic areas. And it was an incredibly interesting job with a huge learning curve that never really stopped spiking upwards, really. There was always something new happening. And it was a very rewarding and interesting role. And after a few years of doing that, I moved onwards and upwards to be head of epidemiology at the MHRA, looking more at the post-licensing side, which is very interesting, especially as we're doing kind of starting the rolling out of vaccine safety studies. So the experts in my team were looking at that, and these have become much more high profile over the last 2 years, should we say?
Yes. I think the post-marketing authorization work, also from the regulatory side, has much more kind of visibility nowadays than it has in the past, for sure. Yeah.
Yeah, definitely. And I think that leads on nicely to where I went next, which is EMA, who've been very much integral in making sure that visibility is there. And so I've been at the EMA for about 7 years now. A lot of the work I do is internal consultancy from a statistical point of view, and also finding ways to go forward when there are perhaps differences of opinion between different member states. We still have to have one unified position. So finding the optimal scientific route for that is one of my key roles. I sit in the task force dedicated to data analytics and methodology, which has been going a couple of years now, and it's a real hub for quantitative methods at EMA.
Yeah.
I started getting involved in extrapolation actually about 6 years ago, I'd say, when the reflection paper was being drafted. And it's very much a kind of cross collaboration between clinicians and statisticians and pharmacometricians and modelers in particular. So to bring those three disciplines together and to get everyone at the same table and singing from the same hymn sheet, I helped steer that through. And and so I'm now on ICH E11A as well, which is paediatric extrapolation. I'll talk a bit more about that later. That's out very soon, and I lead the stats subteam on that.
Yeah, very, very good. I think this is a really, really important topic because I myself have 3 little kids, and I know that for lots of different compounds, there's very, very limited data for children. And so, but still, of course, children need to be treated. And I think there's a lot of physicians that need to treat or pediatricians that need to treat patients irrespective of whether there's an approved product or whatever. And I really rather have them some really good, solid data and quantitative assessment than, just maybe some anecdotal things and just personal opinion.
Yeah, and I think that's very on point. At a holistic level, extrapolation is about bringing drugs on label so that all prescribers have information on safe and effective doses in the paediatric population. So it's not just those who have used it off label and those who are happy doing so, but every treating physician should have the evidence that they need. Yeah,
it's really about making data available for good decision making in the end, and that's one of my personal kind of goals to improve that. So when we speak about extrapolation, what actually is that? Because when I'm just thinking back of my stats parts and I think, okay, maybe you have a linear regression and kind of most of your observations are between, 50 and 150. And then you try to extrapolate what happens beyond 200 or something like this.
Yeah.
Is that the same thing?
Well, not exactly, no. But I think, maybe the principle is the same in that we want to try and bring the label down from adults to include younger age groups in it. But I mean, we define it quite clearly as being, it's based on information in one or more source populations, such as adults or children, that's relevant to a target population. So, for example, another paediatric population, in a way that can be quantified and used as a basis for further development. And so effectively that's making use of information in one population to guide the design and analysis and interpretation in another. And we've actually phrased a lot of our reflection paper, it's age neutral. We don't talk necessarily about paediatric extrapolation because it applies across development where you could have such populations. Now, it just so happens that paediatrics is perhaps an obvious main use case because you have this source population of adults and you have a target population of children, and you know that you want to be able to do as rational development as possible. So it's the principles apply across development, but the key use case right now is paediatric.
Okay, so it could also apply, for example, older population. It could apply for kind of potentially patients that, were previously excluded from studies for whatever reasons, these kind of things. Yeah.
Yeah, exactly. And there are indications where we have used extrapolation for that because of, as you say, patients who've been excluded. Male breast cancer is an obvious example. So yeah, it's something that that can work. But I think perhaps the difference is that PEDS is often after adults. And I think that's often as it should be in terms of development. We all want safe and effective medicines for children, absolutely. But if we have no real evidence that it's safe and effective in adults, then I do think people start getting nervous about whether we should be starting straight away in the paediatric population. So it is kind of inevitable you have this order to development in many situations.
Yeah, absolutely. I think it's the same what we have seen with COVID and then in the pandemics that, first it was for adults and then you went into more and more younger patient populations. Yeah. So in the document, and I'm referencing this in the show notes, it says a lot about talking about the source and the target population. So can you speak a little bit to what that exactly is?
Yeah, so the source population is what you are extrapolating from, and the target is who you are extrapolating to. And so we would normally think, so therefore, that if we're talking about pediatric extrapolation, that your target population is some sort of pediatric population.
Does that mean that we have no data whatsoever for the target population?
No, it doesn't. It doesn't at all, actually. And I think that the purpose of this is to work out what data we need based on what we have already. So your sources might be adults, so you could extrapolate from adults to children. But children are not homogenous, and they're not just small adults. They have different organ maturation at various stages. And so the extrapolation from those different age groups can be different. So you could, for example, extrapolate from adults to adolescents, and then you could extrapolate from adolescents to 6 to 12-year-olds, and then extrapolate from 6 and 12 down to 4 and 6 and 2 and 4. In each of those situations, you would have some information in a relevant pediatric population of some sort, which you could then use to inform what other information you would need in those different age ranges that you're looking into. Okay.
So is it all kind of safety, efficacy, and pharmacology data, or do you also use other data sources?
Well, in our reflection paper, I'll talk about the extrapolation concept and the plan first. So we've split extrapolation into two main steps. On a more technical level, we have the extrapolation concept and the plan. And the concept is kind of held up by three pillars of pharmacokinetics and disease and clinical pharmacology. So the drug, the disease, and the drug and the disease. And we have a table in our reflection paper. And I think it's not a box ticking exercise. It's a box filling exercise.
Okay.
And we need to identify where the gaps are in the knowledge. So, for example, we might know a lot about clinical pharmacology in adults, which we probably should do by the time that we're embarking on pediatric extrapolation. We should know about the natural course of disease in adults as well. That may be different in children, but we should find the evidence to do that. So, for example, real-world evidence data sources may be able to tell you something beyond what clinical trials show, to show that actually the course of disease or the time taken to subsequent therapy or whatever is the information you're looking for may be available in real-world data sources as well. And of course with the preclinical models as well for the ADME data. So there's all sorts of possible data that could be brought to the table to fill in these gaps in the knowledge. And to me, that's why it's quite an interesting area to be involved in, because you can work with many different data sources and types and experts in the field.
That's cool. Yeah. I completely see that. It's kind of pulling data from all the different sorts: clinical, epidemiologically, real-world evidence data, pharmacokinetic and PK/PD data, and combine them all into one kind of model, one kind of concept or framework is quite interesting.
Yeah, it's quite hard as well, and I don't think that should be underestimated. I think so. What we're trying to do is to say, how much can we trust the data in the adult population as being relevant for the pediatric population we're interested in? So the adult data is what it is, and the studies have been done, and we have that information. But then the work on the ClinPharm and the disease similarity and the ADME data all adds together to say, and because of all of this additional knowledge, this is how much we can trust the adult data. And I think that it's a knowledge synthesis process, and also a large degree of clinical judgment as well.
Okay. So in the guidance, it states, where possible, quantitative methods should be used for the collation of available data and the investigation of potential modifiers of the treatment effect. And I think in terms of modifiers of the treatment effect is really about kind of the differences between the target and the source population, what we are mostly interested in, not other modifiers.
Yeah, I think that's a correct interpretation. And I think in terms of quantitative methods, well, the first thing that springs to mind is meta-analysis. It's a standard part of the tools. But we also have, our pharmacometric colleagues. They're very good at dose exposure modeling and exposure response modeling. And all of those will be potentially different in adults and children. And I think what we would like to do is to link that together with better predictions for efficacy, because we can accept that there may well be different efficacy in children compared to adults. And that's okay, because as long as we know what that is and we've quantified it and we've got data to robustly confirm that, then we don't necessarily need to demonstrate, have a standalone demonstration of efficacy as it is. Or perhaps maybe we need it to confirm that our modeling is sufficiently robust. And I think it's important here to recognize that we don't have equipoise per se, because we have the adult data where something has been demonstrated, but we're not operating in a vacuum. And the requirements for what's required for paediatric development will have to take that information into account. And so if we're not demonstrating efficacy, but instead confirming that predictive efficacy is as we expected, then that will require a different amount of evidence to demonstrate that. And I think from a kind of a practical point of view, that, or theoretically, a bit of both really, it always needs to be less, because otherwise we're not doing extrapolation. If we do this whole exercise and do lots of modelling and then conclude that we need more data than we would have done doing standalone efficacy, then something's gone wrong.
Yeah.
And the other thing as well, I think it's important to note that we don't always need data to verify predictions either. Now, the purpose isn't to kind of confirm the model for the sake of it. Now, there's an uncertainty there we don't know, and we need the data to be convinced. But there are situations, so certain anti-infectives, what you're trying to do is you're trying to get a sufficient amount into the bloodstream to kill the bug. It's not about generating a receptor-driven response, for example. So as long as we know what the PK is and we have a safe dose that we can use in pediatrics, then we don't necessarily need any additional efficacy data depending on the situation.
So that's exactly where kind of the disease comes into play. The better we understand the disease, the better we can, be sure that there's certain kind of relationships hold true.
Yeah, very much so. And I think, there's been huge advances in many areas where we've got a really good understanding of the disease, and the understanding has improved, and that's driven drug development. Kind of immunological challenges in children, for example, in ulcerative colitis and pediatric Crohn's and things like that. We have a wealth of products compared to what we had 20 years ago, and a huge more understanding of the immune-driven responses of the disease and the drugs used to target it.
So if I'm thinking about the data from the source population, do I need to kind of treat different parts of the data with different kind of weights, so to say, or how much it helps me? So just if I'm thinking about, let's say I want to extract to kind of teenagers and I have a study, above 18 years old, is the data kind of that I collect between 18 and 25, is that potentially more relevant than, let's say, the data of those being over 50?
Yes, it is. And I think that is a challenge that we have, is that companies can't always control precisely the age distribution of people who go into their adult development programs. But I think we all, when looking at, say, extrapolating from adults to adolescents, would think it is more relevant. I think, again, where statisticians would need to get involved would be appropriately designed subgroup analyses and interpreting them, because ultimately, if you don't really see a difference in the age subgroups, that perhaps lends not weight that extrapolation is more possible or is a better option, but that you could use the entire adult data set and be confident using the adult data set and not just the 18 to 24-year-olds. But I think we do start at the default position that some data are more relevant than others,
Yeah, I see that. And so that's actually quite interesting also if you kind of plan for going into more younger populations. having potentially more patients on the lower end of the spectrum could help you to gather more data in that regard, yeah?
Yeah, definitely. And I think there is one of the challenges that exists, I think, is encouraging companies to design adult development with an eye on the inevitable pediatric development that will come afterwards. We make companies agree pediatric development plans after adult clinical pharmacology studies, often paraphrased to at the end of phase 1, but there's sometimes a little more nuance than that. But we would like to be able to make sure that the trials would be relevant so we can utilize as much. And yes, that's about age distribution. And sometimes it's about things like collecting relevant, important biomarkers or something similar, which companies might be thinking about doing anyway. But if we have a conversation, we're saying this will be really crucial to facilitating an expedited pediatric extrapolation plan. Hopefully, we can encourage people to think about the design of the adult studies with more than just one eye on the pediatrics.
Yeah, that's really good. I think I see it also from a kind of HTA perspective. There's much more kind of we can do with good phase 3 package than just getting regulatory approval for the adults, which of course, it's also quite a hurdle to jump over anyway. But yeah, that's really good. What further challenges do you see? What are kind of your biggest concerns that you have?
Well, I think they kind of fall into many different areas, and there's the logistical challenges of extrapolation in the timings of this. As I've said, you're supposed to agree your pediatric development plan at the end of, including the sample size, at the end of phase 1 in adults. And the timings of the interactions with the regulatory authorities is difficult because what we've said in the reflection paper, we'd like to be as iterative as possible because we recognize and accept that new data comes out through the adult development, which will influence the pediatric development. But also what we would like to do is to minimize the amount of modifications that companies have to make to their pediatric plans as well. We'd like to be able to say, well, yes, let everything go smoothly. And if it all happens as you hope it will, then carry on. And if not, come back and talk to us later. But finding words to do that to fit with our legislation just involves a lot of upfront planning. And it is hard, but we would like to agree as much as possible, as early as possible. I think also that pediatric development is global, and it almost inevitably is. And that means that companies will have to agree across agencies globally. You have to get agreement between different EU committees, such as the Paediatric Committee and our CHMP, our licensure, but also between EU and FDA and indeed beyond. So ICH 11A hopefully will go some way to ensuring that that's going to be more straightforward in the future for companies.
So this kind of pre-specification thing or kind of agreeing to things before we collect the data, I think is one of the paradigms that we have in drug development to kind of avoid fishing for significance and these kind of things. I think that is particularly, difficult for these models because you pull data from different sites and you have the different studies coming in at different time points. Is that kind of the background for the pre-specification and kind of this iterative approach?
Well, I think if we're going to agree a pediatric sample size at the end of phase 1 in adults, and then we have phase 2 and phase 3 data, which don't necessarily line up exactly as the company thought they would in either direction, both impressively or less impressively, then I think that information will need to be taken account into the pediatric development. And obviously, if you have a drug that turns out to be an absolute wonder drug and has a huge treatment effect, and there's good evidence that the mechanism of action is going to be similar, at least in some pediatric subgroups, then we will be more comfortable saying it's reasonable to conclude that the benefit risk is positive in children. Whereas if you've got a phase 3 package where you didn't get as big treatment effects as you hoped for and you just squeaked over the line with P equals 0.049 or whatever it is, then it's inevitable that the situation of how comfortable we would be extrapolating and thus how much data or what type of data is going to change. And I think also that's the other main challenges are the design and the analysis of these studies.
Yeah.
And the design is at quite high level whether you would require a randomized controlled trial but at a different level of evidence, or if you would require a single-arm trial, or if you would require something that could rely on only on pharmacokinetic data because you've got sufficient.
Yeah, I think that's a very good point. In the end, it's about how comfortable are you with the decision making, and the overall robustness of the data is important.
And we do talk about extrapolation being a spectrum, and I think we've moved away from decision trees or ways of saying, if this, then that. But nevertheless, there are a finite number of study designs available to developers. And so working out, given all of this information and all the uncertainties, which are the trial designs that we want is part of one of the main challenges. I think another one which needs to be mentioned is kind of Bayesian and frequentist designs. And I think it's potentially a very rich area for the use of Bayesian statistics, because you have some sort of prior in the adult's data, because you do have prior information. And when people hear the words prior information, it's only natural, I think, that people think, that means I should be doing Bayesian statistics.
Yeah.
And indeed, FDA have been driven by PDUFA 6, where their legislation says they have to investigate Bayesian methods, and it's an obvious place to start, really, because of this prior information. I think we're still not entirely sure of the utility of all Bayesian methods, and that they're not all created equally.
I think the one thing where you get the information is, of course, from the adult population. The other thing where you get information, especially about kind of standard of care or placebo treatment, is, of course, real-world evidence data. Yeah. And so that's another piece of the puzzle that you can fit in. And I just recently recorded a podcast about how you can use real-world evidence data to kind of help with your comparison data for a clinical trial. And isn't that going into a similar direction?
Yeah, it is. And I think we tend to use the phrase augmented, I think is probably the buzzword at the moment that we would probably use. That augmenting the control arm, potentially using other randomized control arms is one way, but sometimes they don't exist, or you don't have anything that you think is actually relevant. And so you might be able to use that. I mean, if you want to use a successful randomized control trial arm of an active and show superiority against that, I think we might be quite happy with that. But it's
at least on the efficacy side,
And so there will be situations where real world evidence is suitable for that, and it will depend on the design and the disease and the indication and all of the things that go into these thinkings in adults apply in this situation as well. But it's clear that the door is not closed here, and that there are options available. There are differences of opinion between some regulatory statisticians about whether it would be better to borrow the treatment effect from adults, or whether you would prefer to just augment a control arm. And I don't think there is a unified position as to which of those two approaches would be preferable. And there are strong views on both sides of the argument on that.
Yeah, I can foresee that this is not a closed discussion that could kind of go on for quite some time. Maybe we can kind of at one time come up with some kind of unified approach there that takes both sides into account.
Well, it's a possibility, and I think that's the good thing about well thought out Bayesian designs. For example, if you're not making multiple changes and you're not having multiple interim analyses, and actually it's quite a clean Bayesian design, so to speak, but you just wish to interpret the data in light of the prior, then you can run a paediatric program successfully and analyze it in a frequentist fashion, analyze it in a Bayesian fashion, and come to conclusions based on that. And indeed, if you read the assessment reports from the FDA for belimumab in lupus, they chose a Bayesian method as one way of characterizing the efficacy. And in the EU, we chose a more frequentist way to interpret the data. But the key point was that because the trial had been sufficiently well designed in the first place, both interpretations were able to happen. And it was all a little bit on the post hoc side, and it's not necessarily the poster child example one might use, but it is a good example of different regulatory authorities applying different frameworks for analysis and still coming to similar conclusions.
Yeah, I think that is actually quite nice. If you have different analysis approaches and vastly different conclusions, I'd always be concerned anyway. So that's a good point. Any other kind of big challenge that you see kind of beyond the things that we talked about in terms of analysis and logistics?
Yeah, I think, again, the design measuring what you need in the pediatric trials, in the adult trials, is something that needs to be really thought out. But another really big one is communication. And that's communication between companies and regulators, and also between other stakeholders as well. And again, we need to be clearer regarding the acceptability of certain designs, in particular with Bayesian ones, the operating characteristics and the type 1 error and the type both conditional and unconditional, and what the important metrics are for success for regulators. Because if we don't tell you in advance what our metric for success is, then it's quite difficult for a company to go away and, in particular, to power and to size their study. Yeah. So we do want to know properly what these operating characteristics are, and if companies say it will be this, and we can accept that. I think some of the more technical Bayesian designs, such as dynamic borrowing, where we don't know in advance how much borrowing is going to take place because it depends how well the data matches the prior. Things like that, certainly we understand the models, but we're just not entirely sure that they routinely, robustly provide all the outputs we want if all the borrowings is left to the model. So we need to perhaps better communicate between industry and regulators as to what our standards and expectations are. And then once these have been successful, to kind of tell the outside world that, because some people will inevitably look at a trial and say, oh, that had a p-value of 0.1, so therefore it's a failed study. And that's not how we would interpret it in this situation. And trying to explain to the outside world that from our point of view, actually, given the experimental circumstances and everything else we know, actually that's a success.
Yeah, I think that's a good point, that it becomes much more challenging to kind of communicate well all the evidence together. I think that is also potentially we need to communicate these models also in a different way, rather than, just taking a number like the p-value you just mentioned, but more describe kind of the distribution of things. More describe kind of the relationships, for example, in terms of the treatment effect modifiers. Potentially, that there's no effects there, and to show kind of all the data is really robust. Would you think like there would be potentially some more visual tools to show these kind of things rather than tables? I'm just thinking about kind of predictive analytics and Bayesian analysis. Lots of this is really, really much more kind of about distributions rather than point estimates.
Yeah, very much so. And I think one of the challenges we face with some Bayesian models, for example, is that the unconditional type 1 error can be computationally calculated, which we can accept, and then that can be plotted against various underlying parameters, and it's not constant. And actually understanding where the type 1 error is raised, as well as how much it's raised by using these Bayesian methods, is inherently a visual display. And I think that there was a paper from the DIA Bayes group quite a few years ago now, with Kurt Fieler was on the lead author, and that one's really good for giving examples of how you might display data like that. Other visualizations which I'm particularly fond of are thinking about, what does success look like? And as an example, we will say a company will make a proposal for some analysis plan and say, okay, well, if we were to see, say it's 40 subjects per arm, randomized 1-to-1, and if we were to see 30 subjects successful on control, how many would we need to see successful on active? There'd be 35 to 40 or whatever the numbers are. But then looking at different cut points, if you want to see if it was 10 subjects successful on control, how many? So really understanding when would a company consider this trial as a success? And then we can say, well, actually, that then translates to a certain treatment effect. And we would say, well, yes, that looks like an important treatment effect that we would be interested in. And if you can go through that process, then you can agree a program and a sample size and success criteria quite quickly. And I do think that better visualizations for that certainly help focus the mind in terms of that relationship between the size of the treatment effect and the sample size you've got to hand.
Yeah, that's a very, very good point. You mentioned this lupus case study.
Yeah.
I guess that is publicly available, isn't it?
So there's always a wealth of information on regulatory websites. You can look at the EU, the European Public Assessment Report, and the FDA information is available on their website as well. And indeed, I think the regulatory websites are a very good resource for that kind of thing, because sometimes it's difficult to communicate to the outside world. It's not necessarily something that people would write up in a paper for the outside world. Actually, some of the most interesting extrapolation case studies I've seen are ones that have been presented at conferences. And one of the things that's happened for the pandemic is I've been able to at least online attend a lot more of these meetings than I have been previously when they might have been face-to-face. There's something that's so great about the face-to-face approach that I'm hoping is going to be returning very soon. But certainly I've been exposed to a much wider array of things like case studies, because these are perhaps the fora that people, webinars and so on and so forth.
Okay, let's put a couple of these into the online appendix here, our show notes. And then you as a listener, you can have a look there. Just head over to theeffectivestatistician.com and then find this nice episode with Andrew. Thanks so much, Andrew. That was an awesome discussion. We started really with kind of what actually is extrapolation, and that we really want to extrapolate from the source to the target population. We had a lot of discussion about what type of evidence we need and where this can all come from, and that it comes from disease understanding, it comes from efficacy and safety data, it comes from PK data, but it can also come from real-world evidence data and other data sources that help us to have a good understanding of the overall data to make a good recommendation for physicians, for patients. And we also talked about, challenges like communication, logistics, having good kind of iterative planning on terms of all the data that comes in, and whether that kind of changes our plans, or how we can, make sure that we have all our plans well thought through. And if there's some change in the data that we didn't expect, that we can make amendments, make things faster or slower, depending on the data. Is there any final thoughts that you would like to leave the listener with?
I just want to mention ICHE 11A, which should be out very soon. Hopefully, by the time some people are downloading it, it may already be out. So that'll be out for consultation. So please do comment on that and help us improve the document. And also, finally, thank you for inviting me. It's been very nice to talk to you.
Thanks so much. Did you enjoy this episode as much as I really enjoyed the discussion with Andrew? Then let me know about it. Connect with me on LinkedIn and tell me what you think. This show was created in association with PSI. Thanks to Rain, who helps with the show in the background, and thank you for listening. Reach your potential, lead great science, and serve patients. Just be an effective statistician.